Hi, everyone. Thank you for inviting or letting me give a talk here. Here to talk about some work that I've done at GFDL on global scale ecological and biogeochemical impacts of pelagic tunicates. And this work was done with collaborators Charlie Stock, John Dunn, Natasha Hensky at University UNSW, New South Wales, and Todd O'Brien at NIMS S&T. So gelatinous zooplankton are a diverse class that are found all throughout the world's oceans, ranging from the large scyphozoans to comb jellies, these ctenophores, various types of hydrozoans, and then the key type of organism within that I'm talking about today is the pelagic tunicates, and that's a SALP up there. They're characterized by these very fragile watery bodies with low carbon content, and as a result, they're able to achieve a very large size without the associated carbon content or metabolic rates. They're very effective at passive feeding, and many species produce asexually these very large blooms, which David showed earlier today. So there's been a number of historical barriers in studying gelatinous zooplankton, and I'll highlight a few. Originally, they were thought to be trophic dead ends, but because of more recent technologies relating to stable isotopes, DNA barcoding, animal-borne cameras, there's evidence showing that many types of organisms throughout the world's oceans, including top predators, predate on gelatinous zooplankton, including pelagic tunicates. And then also they're associated with this, there's been kind of a general unknown sense of their importance within marine ecosystems and biogeochemical cycles. And then lastly, they're very difficult to sample with traditional methods. So very notably, historically we sample plankton with these net-based systems, but they're very biased for gelatinous zooplankton. So in this study from Andrew Remsen et al. 2004, he compared side by side a net-based imager with a net-based system with a plankton imager, and found that, well, you can see, I don't have a pointer here, but these panels on the right, for the crustaceans, the nets and the imagers matched pretty well. But for gelatinous or other soft body organisms, there was quite a big difference between the imaging systems and the nets, such that, for these appendicularians or larvaceans and pelagic tunicates, in terms of carbon, their difference was, between 5 to 15-fold difference. So I think that we've been generally underestimating their biomass within the global oceans. And then also among gelatinous zooplankton, it's been thought that pelagic tunicates in particular contribute disproportionately to biogeochemical cycles through their fecal pellets and these like jelly falls, these mass depositions of carcasses on the seafloor. So in this study that was published in 2020, we did an offline modeling study where we estimated based on data what the presumptive flows of carbon could be through the gelatinous zooplankton. And so here in this schematic, I'm showing just the amount of carbon that's coming in all units of petagrams carbon per year through, for the tunicates in particular, when they're consuming prey, and then a certain amount gets assimilated, and then a certain amount goes to dissolved pools and to predators, and then to particulate organic carbon. Then it kind of sinks to the seafloor. But notably, amongst all the gelatinous zooplankton, even though the tunicates were an order of magnitude lower biomass than the other large jellies, they contributed about 77% of the export. So we think that they might be the most, amongst the gelatinous zooplankton, the most important to focus on. But a key caveat here is that offline models really don't take into account biogeochemical feedbacks. So these values are very high. It's basically suggesting that there's, between 3.5 to 4.5 petagrams of carbon of a particular organic matter produced by these tunicates. So is this actually realistic? When we put this into a biogeochemical model, do we get similar results? So in this study, we decided to put in these gelatinous zooplankton into cobalt. And cobalt is a ocean biogeochemical model of intermediate complexity. Here is a schematic from Stock et al. 2020. It models a heterotrophic bacteria and 6 different types of plankton, 3 sizes of phytoplankton and 3 sizes of zooplankton. Over there. And we thought that to focus on pelagic tunicates, there is one particular aspect of their ecology that is quite important, is their feeding preference. So in this figure on the right, from Conley et al. 2018, this shows the predator-prey length compared with the clearance rates for the pelagic tunicates. Can you see my mouse? No, you cannot. The pelagic tunicates on the top, the pyrosomes, appendicularians, doliolids, salps, compared to the copepods, ciliates, and flagellates. And what you notice immediately is that the pelagic tunicates have among the widest predator to prey size ratios within the ocean. So we think that they are really important in taking, prey from within the smaller size classes and transforming it to particular organic matter. We had 4 major scientific questions that we're interested in. The first, if we're accounting for systematic undersampling by nets, are we able to capture large-scale gradients in pelagic tunicate biomass? And then second, can an expanded role for gelatinous zooplankton be reconciled with established evidence for just the importance of crustaceans within marine ecosystems? And then third, how do the online estimates compare with kind of the previous offline studies? And then lastly, what is the impact of the gelatinous zooplankton on the partitioning of carbon flows to recycling, export, and flux to higher trophic levels? So here's a schematic of the base cobalt food web compared to what we added into, now I'm calling GZ cobalt. So again, there's 3 sizes of phytoplankton, 3 sizes of zooplankton, and unresolved higher trophic level predators. I won't go into the nitty gritty details here of the food web, but in short, the prey preferences of the small tunicates and the large tunicates are actually quite similar. The large tunicates, despite being much larger, have a larger predator to prey feeding preference. So they're able to feed on similar sized organisms as the small tunicates. The distinction between the small and large, perhaps, is that the small tunicates have a higher predator, kind of a predation pressure upon them. And I considered, this is my trait slide. I considered these trait trade-offs for pelagic tunicates compared to the crustacean zooplankton. So I'll kind of walk you through these. The first is the multiphagy. The pelagic tunicates are able to predate on a wide range of organisms, including picoplankton, compared to the crustaceans, who just target primarily on large phytoplankton and other smaller zooplankton. They have this very wide predator to prey size ratio. They have higher clearance rates. And then as the trade-off, they have very high basal respiration rates. They're continuously filtering, so they basically have to maintain a very large metabolic rate, just even when there's very little food around. But as a result, we set it so that they have a relatively lower active respiration rate, so less respiration as they're digesting food. One of the notable things about the tunicates is that there's the clogging of their filters and their house, because they're filter feeders at very high food concentrations. And so what we did, that how we modeled that is that we made the assimilation efficiency decrease with increased food concentrations. And I'll show you a little bit of that with some plots in a little bit. And then I added in this aggregation mortality, this kind of mass die-offs under food depletion. This kind of represents the mass jelly falls of the South. So here are just some like idealized plots of the ingestion, assimilation efficiency, and then specific production at just 25 degrees Celsius. So starting from the top left, the dashed lines are the kind of the existing zooplankton within cobalt. So the top one is the microzooplankton, and then two sizes of the crustacean zooplankton. And then the two blue lines are the small and large tunicates. So in terms of ingestion, the tunicates kind of fall in between the various different zooplankton types. And then you can see kind of the effects of the assimilation efficiency on plot B. And then plot C is just the specific production as prey biomass increases. So then you'll see kind of the effect of the basal respiration rate, which kind of just brings the entire relationship down a little bit, and combined with the decreased assimilation efficiency. All right. So we compiled for this some observational validation data from a range of data sets, including the NOAA copepod data set, the Jellyfish Database Initiative, and Krillbase from the Southern Ocean. And so you can see just overall kind of data scarcity throughout the world's oceans. And then most notably, as we started pulling together the data, we noticed that there was a contrast between the gelatinous organisms versus the crustaceans in their relationship to chlorophyll. So if you look at this plot in the center, we noticed that particularly the large tunicates really just didn't have a strong relationship to chlorophyll, that you can see very distinctly in the crustaceans, the large mesozooplankton. So this kind of suggests that as climate warms, as chlorophyll may decrease, the relative importance of tunicates may increase. There just may be more room for them within ecosystems. And then the small tunicates kind of fell in between. And you can also see, these are on log scale, so you can see just the widespread in the data. It's extremely patchy. All right. So we created this simulation. We ran it for 60 years on a coupled physical biogeochemical model, MOM6 and GZ cobalt, using this core 2 interannual forcing. And everything that I'm showing is the climatology of the last 20 years of the simulation. So what you can see here is the overall, you have this, we're seeing overall kind of very much less distinction between the biomes in the large tunicates versus the crustaceans, kind of consistent with the data constraints that I showed a little bit. And I'll show you the comparison on this biomass chlorophyll space in a little bit. And then the small tunicates were getting this much lower biomass. They're about an order of magnitude lower than the large tunicates and crustaceans. And here I'm showing, again, this biomass chlorophyll constraint. We're kind of calling this not an emergent constraint, but an emergent relationship within the data. And the black dots are the model. And so we were able to largely capture the difference between the biomass, the relative difference between the biomass of the small tunicates versus the large tunicates and the crustaceans, as well as this contrast in this relationship with chlorophyll, which this ended up being one of the primary validation metrics that we used. In terms of the seasonal cycle, and now what I'm showing you is this adjusted seasonal cycle where the southern hemisphere is kind of flipped. So the summer months are June, July, and August, are basically months 6 through 8. And I've divided the ocean into these 3 major biomes: the low chlorophyll, which is in green. Apologies to those who are colorblind. The blue is the upwelling high chlorophyll zones, and the orange are the the high chlorophyll seasonal cycle zones. And the dashed lines are the cobalt base simulation, and then the solid lines are the new GZ cobalt simulation. So what we saw is that the introduction of the GZ reduces other zooplankton biomass generally. But in particular, the biggest difference was right here in this upwelling region, particularly for the small zooplankton, the microzooplankton. The large tunicates were notable in that their biomass was highest in the upwelling region compared to like in the high latitude seasonal cycle seas. And this was in contrast with the small tunicates. But instead of looking at just individual differences between specific zooplankton groups, I thought it would be more illustrative to look at these patterns in terms of the processes that go to recycling versus the processes that go to export. All right, so what I've divided up here in these plots are processes that go to recycling. So for example, the ratio between microzooplankton production and MPP on the top row. And then the second row is these general process that goes to export and higher trophic levels. So this is all mesozooplankton together, divided by MPP. And then the third row is export flux over MPP. And then lastly, it's bacterial production over MPP. And this is a process that goes towards recycling. And the left-hand column are the GZ simulations. The center is the control. And then the right is the difference between the two. And what you'll notice immediately is that overall, the introduction of the GZ into cobalt results in decreases in the overall flux to recycling compared to export. There wasn't too much of a latitudinal gradient here, particularly in the bacterial production decline. There really wasn't that much of a difference. And with the PE ratio, just the amount of export flux divided by MPP, the differences were higher in the higher latitudes, for sure. But really, there wasn't too much of a latitudinal gradient. And then when I look at the contribution to export from the different groups in the control versus the GZ cobalt, the first thing that I noticed is that the overall export production in the top 100 meters doesn't actually change all that much. The difference is about 1.1 petagrams of carbon per year. But most notably, the introduction of the tunicates redistributes the export production. So the small mesozooplankton are decreased by 30% or so, and the large phytoplankton are also decreased. And in comparison to the offline model, I have the numbers on the bottom right there. The offline model suggested that it was around 3.3 to 4.5 petagrams of carbon per year. And here we have about 2.7 petagrams of carbon per year. So it's slightly on the lower side compared to the offline model. And then this is the flux to the higher trophic levels. So this will be like the fish and the squids in the model. So in the introduction of the GZ cobalt does, again, decreases the fluxes to the higher trophic levels, but not by too much. It's about like 0.3 petagrams of carbon per year. This is much lower than what the offline model suggests, but perhaps we're just not including enough predation on these tunicates. All right, so my concluding points. So many empirical studies hint at this expanded role for gelatinous zooplankton in marine food webs. And we were able to create a simulation that we're able to capture these large-scale emergent patterns in their distribution while accounting for this systematic undersampling. The simulation satisfies a range of biogeochemical constraints. I didn't show you here in this talk, but when you compare the surface nutrient concentrations, the new model is quite skillful and really is not that different from the control. But I was also able to maintain a very robust population of crustacean zooplankton. The pelagic tunicates are in significant competition with the microzooplankton, and this is consistent with results from the Southern Ocean that show that the salps in the areas compete very strongly with the microzooplankton. And in the model, this was most important in the upwelling biomes. The net biogeochemical impact of these organisms was to shift the system away from recycling and towards export. And this came from a repartitioning of the export from other sources. So we can't really think about these gelatinous zooplankton as organisms that we didn't account for in the model. So once we add it, it'll increase export by a huge amount. But rather, once we add these organisms in, they result in a kind of a reshuffling of the plankton ecosystem. So yeah, that concludes my talk. Thank you very much. I'll take any questions here.
There we go. Any questions, online or offline?
Jessica, correct me if I'm wrong if I didn't read your maps right, but it looked like the large tunicates lived kind of everywhere, and then the small ones were focused in the subtropics principally.
Well, so yeah, the large tunicates, they do live everywhere. The small tunicates were more prevalent in the kind of upwelling zones in the higher latitudes and not in the subtropics.
Okay. I was just wondering if you could explain that, because I think for crustacean copepods, I would expect almost the opposite pattern.
Yeah, so it arises in the, I'm not entirely sure from, if the model processes are fully capturing what's going on in the real world. But in the model, what's going on is that the very high basal respiration rates and the precluding the small tunicates from the subtropics. Their basal respiration rates were roughly like 5 times greater than that of small crustaceans. So even though they're able to consume, they're kind of like filter feeders, so they're kind of vacuuming everything in their midst. But because of their needs to just keep beating their tail to keep that water filtering, it ends up making it so that it's energetically not as favorable to be in the subtropics. Yeah, and like the large tunicates are able to eat the same prey, but they don't have as high of a basal need.
Any other online question? Oh.
It looked like, can you hear me? It looked like in the comparison that you made between the model and the data, it looked like there was a much higher variance in the data.
Yeah.
I was just curious what you thought. Do you have a sense of why that might be, or?
So there's a couple different elements of it, is that there hasn't really been systematic sampling of these organisms in oceanographic studies. A lot of times the oceanographic plankton work will focus on the zooplankton work will focus on crustacean zooplankton, copepods, krill, and other hard body organisms. We did make an adjustment for the net sampling bias, but that adjustment may not have captured enough of the biomass, kind of the bias in capture, particularly for the small tunicates. So the small tunicates are quite tiny. I've read that they're best captured with a 50 micron net, for example. And most of the data that we pulled from were using 350 micron nets. So there's I think that, depending on the type of net that somebody will be using, there could be a lot of variance associated with that. Another source of variance is just the life history. So the large tunicates, the salps, kind of have this split life cycle where there's an aggregate form and a solitary form, and the aggregate form will reproduce asexually. And they split basically. In observations, you'll find kind of either like a large amount of the aggregate forms or primarily solitary forms, though sometimes there will be some mixing. And we did not explicitly represent that diverse life cycle here, partially because we were hoping that we could just capture the mean state with kind of more of a more traditional zooplankton type modeling. But yeah, that could also be a source of the variability. And, as with any other modeling, there could be variability associated with small scale oceanographic features.
I actually had a question regarding the modeling, because when I think of those gelatinous things, they're beautiful but bizarre. And so were there fundamental differences in the way you modeled them? Like you showed some of it, but I didn't get the whole aspect of it. So larger prey selection or other aspects? Or?
The biggest difference, I would say, out of all the things I put in, were the larger predator to prey size ratio. So they kind of break the traditional size trait axes that we think of for plankton. So that was one of the big things, and also their basal respiration rates were just so much higher than other zooplankton, ends up driving a lot of their distribution. Yeah.
Any other questions?
I have one. It's Martina Dolan from Sydney, Australia. Thanks very much, Jessica. Is it okay to ask the question?
Yeah.
Go ahead.
Yep, I just put it in the chat. So although you didn't describe the impacts on the chlorophyll or the phytoplankton group, I'm interested in the effect of the feeding ecology through that very large size range, predator to prey in tunicates. So can you comment on what the effect is on the functional distribution of phytoplankton as a result of this feeding?
Yeah.
Do they make any impact, really, is the question.
Yeah, so they had I didn't show it here. I think there was a bigger impact on the small phytoplankton because the prey preference for both small and large tunicates were on like the picoplankton, basically the picoplankton size range. So it had a slightly greater effect on the small phytoplankton.
Thank you.
Did I answer your question?
Yes, thank you.
And we have an online question by Karen. Can you unmute and ask your question, please?
Sorry, yes, I can ask my question. Can you hear me?
Yes.
Okay. Jess, I was curious, in addition to competition with microzooplankton, do the large tunicates also consume them in your model?
Yeah, they do.
And is that, sorry, go ahead.
So I added, so they have this very wide prey preference, but they're not as good at getting their prey compared to other crustacean zooplankton. So how I did this is I decreased their prey preference or the prey availability of the prey to these tunicates. But yes, they are able to predate on microzooplankton because our microzooplankton in cobalt is also includes some heterotrophic nanoflagellates, and they've been shown to definitely predate on that size class, those organisms.
Are you able to comment on the relative importance between competition and predation? Because you had said that one of the major findings was that competition, there was competition between the tunicates and the microzoa.
Yeah. I don't think I can tell unless I remove that feeding link, and I haven't done that. That's a really good point. Yeah.
Thanks. It was a great talk. Thank you.
All right. Thank you, Jessica.
Okay.